Okay, so the axon hillock, it is this part of the neuron that connects the axon to the actual body. And in this area, that's where all the local membrane potentials add up, and then you can reach a threshold, and then you can initiate your action potential, which is not different than the one that happened in the muscle fibers. So you have to have a depolarization step in which you open your sodium channels, and that brings sodium into the cytoplasm of the cells, and that will create a positive charge inside. Typically, the charge inside these neurons and then the muscle cells is negative, given to the presence of phosphate groups, which has a negative charges, and then that can be reversed with the entry of sodium, which is a positive ion. So then it's necessary to reach a threshold to create an action potential. If that is not reached, then you don't generate an action potential. And then once you generate an action potential, you follow the all or none principle. So you make it or not. And the summation happens in here. So you add any local potentials that derives from a stimuli here in the dendrites, and then this stimuli can be positive or negative depending on the neurotransmitter that this dendrite receives. But ultimately, if you magnify this region and you see the axolemma, remember this is the plasma membrane, and these are the channels that are located in there, sodium and potassium channels. So you receive this local potential, and then you move. This is the graph showing you what happens with the electrical voltage. So this is showing you the voltage from minus 70 to plus 30. So the threshold will be minus 55 millivolts. So if you move from minus 70 to 55, then you reach a threshold, and then all your sodium channels open. And then you depolarize the cell, in this case up to plus 30. And then you reach that peak in voltage, plus 30. You close your sodium channels with using the inactivation gate, so that the pore closes and you don't have more influx of sodium. And then you will open your potassium channels so that you have exit of potassium from the cytoplasm into the extracellular space, and then that returns to the negative value. So your cells will repolarize in this case. And again, your potassium channels, they are slow to close, so that leaves a little window in time for more escaping of potassium. Not always, but when this happens, you have more time for potassium to escape, and then your cells, since they're losing positive ions, they become negative again. So now look at the channels here at this part. So sodium, which is the purple channels, or the channels here seen in purple, they close by using this inactivation gate. And then this will open, you activate your potassium channels, and then you have more efflux of potassium, and then you will return from plus 30 to 0 to minus 40, minus 55, and sometimes again you will have after polarization in which you have more negativity than usual. And then for the propagation, well, the propagation can happen in two ways. By having opening of these sodium channels in between the nodes of Ranvier, and that will give you a saltatory or jumping action potential, which propagates much faster than having continuous propagation of the action potential. So you have some neurons that are myelinated and some that are not myelinated. The ones that they don't have myelin, they have only the flow of the sodium slowly going along the membrane, along the axonal lemma, while in the jumping, it's much faster because you don't have this exposure of the sodium channels where you have the myelin. So, in here, we have an example of what happens internally. So you start opening your sodium channels here, you depolarize your part of the axoplasm in this region, which is proximal, so it will be closer to the body. And then you move these ions inside following the law of opposite attract. So positive will be attracted to negative, and then that will lead to the propagation in a forward direction from the body into the terminals. And as soon as these sodium channels passes from this segment, so into this will become repolarized. And you can tell because here it shows you positive signs or plus signs telling you that this was depolarized, and then in this part where time passes by, you have return of the polarization back to normal, which is negative. And then the next segment depolarizes, then repolarizes, depolarizes, repolarizes, and keep on going until you reach the terminal. Now, when you reach the terminal, in the terminal you have calcium channels. And when these calcium channels open, that will lead for the vesicles that are located inside the terminal to fuse to the membrane of the terminal. And that will lead to exocytosis of these neurotransmitters, and then the neurotransmitters flow towards the plasma membrane of the next cell. The next cell can be another neuron, it can be a gland, or it can be a muscle cell. If it's a muscle cell, you will have the neuromuscular junction, and then you will have contraction. Now, we have within these myelinated neurons also some changes in the speed of propagation. If you have a large diameter neuron and it's myelinated, you will have faster propagation than if you have a medium-sized neuron, and we're talking about the diameter of the axon. So if the diameter of the axon is medium-sized, is myelinated, it's going to propagate less faster than if it's large and is myelinated. The reason for this is because if you have a larger diameter, you have more sodium channels along this plasma membrane. So here then it's an example of the propagation all along the axon. So the axon hillock is also known as trigger zone because this is where you initiate your action potential when you add up all these local potentials that come from these dendrites. And then you generated the action potential. In this case, this is a myelinated axon. So you have jump or saltatory conduction of this action potential until you reach these terminals. And then we have a period of time in action potentials where you cannot elicit a secondary action potential. And this period of time can be subdivided into absolute or partial or relative. And refractory means like you resist. So in this case, you have a period of time where you absolutely do not produce a action potential, so this will be absolute refractory period, or you can have one where you can still produce an action potential if the stimulus is strong enough. That is why it's relative. I always give this example. Well, you have on the weekend an exam coming, and then your friends invite you to a party. But you know that you're not doing well in class and that you better study for that exam, otherwise you will flunk the class if you don't do well. And then your friend insists, oh, let's go to the party, let's go to the party. And then you're a very conscious student and you say, no, I can't. And you don't go. That is your absolute refractory period. But let's say that your friend keeps insisting. It tells you, well, we're going to have a lot of fun, we're going to have this food, we're going to have drinks, and then you're still doubting. I'm thinking, well, that party might not be worth it because I have to spend another semester repeating this class. And then you say, well, no, and then it calls you back again, and then it tells you, oh, guess what? It's going to be the person that you like. As the test. And then you go. That was your relative refractory period. So the stimulus was so strong that you elicit an action potential. So, but then the purpose of having these periods is to allow one-way propagation and limiting the fire frequency. So yeah, it's good to have action potentials, but it's better to space them in time and only when they are needed and only in the right direction. So here we have what happened in each of these periods. There's a reason why these periods exist. So it's not an ordinary fact. So again, we have here the voltage inside the cells from minus 70 to plus 30, and then this is the amount of time that it takes for you to elicit a complete action potential from resting membrane potential up to hyperpolarization. So it takes a little bit over than 2 milliseconds. So how many milliseconds there is in a second? Mil, 1,000. So if, let's say that this one ends in 2 milliseconds, this action potential, how many action potentials can you generate per second? So if every 2 milliseconds you can generate an action potential, how many can you generate in 1,000 milliseconds, which is 1 second? Math Cafe. How many $2 bills can you make out of $1,000? Hmm? 500. So you can generate 500 action potentials in 1 second. How many in 1 minute? 30,000, something like that, right? 500 times 60, something like that. Anyway, so if you subdivide this amount of time, like in here, in resting membrane potential, both channels are closed, sodium and potassium. So nothing happened. Now in absolute, which runs a little bit after time zero, let's say it's 0.015 milliseconds up to 1.75 milliseconds. That's when you have your absolute refractory period. Why? Because in this case, during depolarization, you have all your sodium channels activated. So this won't allow you to activate more because you don't have more available. And then in this case, during this part, it is because you have them all closed. And then this one is open. So this one prevents you then to, this event prevents you from bringing more sodium. But now, basically towards the end of this action potential, when you reach the threshold again at minus 55, some of these are in resting state, and some of these remain activated, but the ones that completed a period of inactivation during this time, during the absolute refractory period, can get ready and you can open more. So then that's what happens with this relative. If you have a strong stimulus, you can still open these ones. And then you have a second action potential generated. Now we have different types of synapses, one that are chemical and other ones that are electrical. In electrical synapses, you transfer ions directly in between cells using little windows in between the cells that we call the gap junctions. We have gap junctions in muscle cells like smooth muscle cells and also cardiac muscle cells. And in this type of muscle cells, it is important to have similar amounts of ions at a particular time so that you can contract all the cells at once so that they can synchronize. So then that leads to rapid signaling. And in chemical synapses, which can be chemical electrical synapses, I guess, you will have transmission of a neurotransmitter in the synaptic cleft, and then your cells will do something. So that chemical signal will be converted into an electrical signal once the cells receive the neurotransmitters. An example of communication. Your mom calls you in the phone and tells you, well, if you are around this supermarket, bring me these items. Milk, eggs, whatever. You go there and you bring them. So it was a verbal signal that it is converted into a physical action. So similarly, our cells send the signals or the message through neurotransmitters, and then the response will be whatever that neurotransmitter opens the channels, what channels are open. And then some of these signals have to be modulated. So you have sometimes other neurons there that can either increase the release of neurotransmitters or the release of neurotransmitters to help to control the message and the action of the cells. So here we have these examples then. So we have two cells in physical contact with each other, and then we have these proteins, which are called connexons, that creates little windows in between the cells so that you have ion flowing. So these are the gap junctions. And then in here, in these chemical synapses, you have your action terminal, you have a space, and then you have this other cell with a plasma membrane that has channels, and then it has receptors for these neurotransmitters that can be released. Now, in this case, the cell that releases the signal releases the neurotransmitters first, this cell will be the presynaptic cell. And the one that receives the neurotransmitter will be the postsynaptic cell. And then there can be some communication back in between the cells. So the postsynaptic can become presynaptic, and this one will become postsynaptic. So, I told you this before, in the synaptic terminals, we have calcium channels that open to receive calcium, and then you can release these neurotransmitters. And depending on the neurotransmitter that you release, is if in your cell, the postsynaptic cell, you will have an excitatory or an inhibitory postsynaptic potential. So excitatory will lead to depolarization of the postsynaptic cell, excitatory signals, excitatory postsynaptic action potentials. And then inhibitory will lead to the opening of chloride channels. Chloride has a negative charge. So when you open chloride channels, chloride comes in, and then that reduces the voltage in the cells. And then don't think that these neurotransmitters are released in the synaptic cleft and they will be acting forever in the cells, in the postsynaptic cells. No, they can be recycled or they can be degraded. This recycling is called reuptake of neurotransmitters. And some part of these neurotransmitters can be recycled, other ones just dissipate or degrades within this space. So here we have the example of then what happens in synaptic transmission. So again, you have an action potential that flows from the body into the synaptic terminal, and then you open your calcium channels, calcium rushes in, your neurotransmitter vesicles fuses to this plasma membrane, and then you exocytose or release these neurotransmitters by diffusion from more to less. The neurotransmitters will migrate from the presynaptic neuron into the postsynaptic neuron, and then you have your channels with receptors for this specific neurotransmitter, and then your ions comes in. And depending again on what type of channel you open is whether you have hyperpolarization of these cells or depolarization of these cells. And if you have these ions in here, that will create a local potential. So here we have a difference between excitatory postsynaptic potential and inhibitory postsynaptic potential in a graph. So if this is the resting membrane potential of a cell, the postsynaptic cell, minus 70, and then you have a neurotransmitter that allows you to have influx of positive ions, that will lead to a slight depolarization, that it doesn't reach the threshold, as you can see here. It can be less than minus 55 millivolts. And then in another area of the cell, or maybe in the same area, but you have other neurotransmitter being released, you will have opening of these channels that are for anions to come in, and then sodium goes out as these negative ions comes in. So that further depolarizes your cells, that goes from minus 70 to minus 80, for instance. And that is why it's called inhibitory postsynaptic potential. That's it. That's a small hyperpolarization. So you end up going to that party. You didn't obey your mom saying, stay, study, going to repeat that class. And then you were all excited, but you didn't reach your threshold because you haven't seen in that party yet the person that you like. But you're still kind of excited there, right? And then that person comes in with a significant other that is not you. So then you hyperpolarize. And you say, I'm supposed to have heard my mom. Because you lose all the hopes. ¿Verdad? So you add everything. So that adding of everything is called summation. It can be in time or space. So temporal summation happens when you have an area where you receive at the same time, at the same synapse, excitatory and inhibitory postsynaptic potentials. So in this case, you add how many do you receive, how many stimuli you receive per minute, for instance. And then spatial. Well, simultaneously in, let's say, 1 second, you receive signal from multiple synapses, and they are excitatory and inhibitory, so you add all these signals. This is a spatial summation. And then, no matter if it's temporal or spatial, you add them all, and then you know if you reach an action potential or not by just having the threshold. So this is what happened with temporal summation. So you have only this area receiving this excitatory action potential, so you add them up, all from this connection. You reach a threshold, you make an action potential. So that's your excitatory that leads to the action potential. And then you have here 3 connections, 3 synapses that at the same time send a signal that leads to the same effect, so reaching the threshold and then an excitatory action potential. Then to follow up with my soap opera, my novella. So then you were at the party all like inhibited because you saw that person with someone else, and then you saw that they fight, and then the other person leaves, and then the other one that you like is still there. You add in time and space, and then you make an action potential. Okay? Now, we have hundreds of neurotransmitters. They have different chemical structures, and then we can classify them based on that. So we have some that are amino acids, like glutamate or gamma aminobutyric acid. We have peptides. We have ATP, which itself adenosine triphosphate can be a neurotransmitter. And then we have biogenic amines like dopamine and serotonin. And some neurotransmitters, they can act in more than one receptor, and that will dictate what response you will have. So it can be excitatory or inhibitory. So we will see an example of this later on. And then again, when it comes to neuromodulation, well, think about all of this. So these chemicals are produced by our body. So it takes energy and resources to form these neurotransmitters. So it doesn't make sense for us to be releasing these neurotransmitters all the time. So then we have to have a sort of neuromodulation to only allow the release of these neurotransmitters when they are needed. So that way we save energy. So now a clinical application of synaptic transmission and toxins. Certain microorganisms or toxins in the environment can lead to the blocking of release of neurotransmitters, and then you can have a consequence. So for example, we have consnel toxins that blocks glutamate receptors, inhibiting the neurotransmission. We have this poison, strychnine, that blocks glycine receptors, causing excessive excitation. And we have some drugs that can affect the reuptake of neurotransmitters or the degradation, leading to increase of these neurotransmitters action and strength, making some effects on the nervous system. Many drugs, they have some warning. It tells you, well, if you're taking these inhibitors of MAO, which is monoamino oxidase enzyme, then you shouldn't be taking this drug because it can potentiate the effect of this and it can lead to problems. Okay, so that was neurotransmission. Now let's talk about functioning of cells as a whole and within groups. So we have neurons that organize into groups or pools that processes a specific information. And they form these groups, but within these groups, you have some cells that communicate with one another in a specific manner, and then you end up with having some circuits that can spread the information. This is called diverging circuits, or some that they can narrow down that information that you receive, and in this case, this will be converging circuits. So in divergent circuits, one neuron stimulates multiple neurons, and in convergent is the opposite. Multiple neurons can stimulate a single neuron. And then we have within these circuits excitatory and inhibitory functions that regulates contraction of the muscles and other functions. So something that can happen, for instance here in the brain, is that we can have a single neuron that can start sending information gradually into more neurons so that you can amplify the recruitment of, in this case, muscle fibers. So this can be, for instance, to control fine movement. A single neuron can diverge the information into multiple neurons. And that happens between the brain and the spinal cord. So you have to send action potentials from this neuron into these neurons of the spinal cord. And then you can have multiple tracks. Like in this case, we have 4 tracks, and they are fed from these two tracks and this one from this single track. So this can happen, for instance, in the neuron in the spinal cord that carries information from peripheral receptors. So this will be kind of an interneuron that relays the information into tracks so that they can reach different areas of the brain to control different functions. In converging circuits, you have input here by multiple receptors, and these receptors send the information into less and less neurons until you reach a particular neuron within the brain. So that's converging. So that's the end of this chapter. In our next chapter, we're going to see specific details about the nervous system, the anatomy and physiology of the brain and spinal cord. We're going to see examples of high-order functions. We're going to see reflexes, how they're performed, and then what do they do in general. And we're going to see some clinical applications and neurological disorders. So test your knowledge questions. What type of neuron carries impulses away from the CNS into the muscles? A, B, C, or D? From CNS into your muscles. Motor. Or efferent. Number 2, which neuroglial cells form the myelin sheath in the central nervous system? Not all at once. That's peripheral.
Oh, okay. B?
Which one? I heard something, but.
I said B, sorry.
Okay, B, yes. I just had to hear well because I heard E and I said there is no E. Yes, B, the oligodendrocytes. True or false? The resting membrane potential is maintained by the sodium-potassium pump and ion leak channels. It's true. Action potentials.
Taking it in and out, right?
Yeah,
So it's
you have,
maintaining it.
basically what you're trying to do is maintain a ratio, more sodium out and more potassium in. And then next one, action potentials decrease in strength as they propagate along the axon. False. Yes, an action potential is an action potential. It reaches the threshold and then the peak in depolarization. Then a third, true or false. Saltatory conduction occurs in unmyelinated axons. And of course, it's saltatory conduction of the action potential.
Is that false?
That's false. Where does it occur?
In
Myelinated axons, where you have the nodes of Rambier. What is the difference between excitatory and inhibitory postsynaptic potential? So which channels open in excitatory and which channels open in inhibitory? Do you open cation channels or anion channels in excitatory? Cation is positive, anion is negative. You open positive. And then what about in inhibitory? You open cation channels or anion channels? Anion channels. So which of the two, excitatory or inhibitory, will lead to an action potential? Excitatory. Okay.